School. Cool. Okay, yeah, thank you so much for having me. Very nice. Yeah, I'm gonna present some work that I did a while ago at the Carnegie Institution for Science, Stanford. What we were trying to do was looking at, or actually I came new into the field of gene regulatory network inference. And when we looked at it, we found we looked at the papers and actually that Dream Challenge paper that was talked about. We looked at this and saw several things pointed out that several things that have been pointed out that were made gene regulatory network inference difficult. So one, for example, being that what is known as the curse of dimensionality problem, that we often have like a large number of genes versus small number of conditions, which is a particular problem if you want to go for condition-specific networks. And then whole kind of different problems when we go from prokaryotes to eukaryotes, that we find that gene expression, gene regulation is also controlled by other factors that is not particularly captured in gene expression data, and this data might be there, might be available, might not be available, or might be partially, so not necessarily genome-wide available. And then one particular thing that I was thinking about is that problem that we often have small gold standards, even for the model organisms, meaning that the gold standard being like experimentally validated regulatory links. And an interesting point that from a machine learning perspective, what we often want to have is true negatives as well, right? So in this case, that would be like absent links, and that's experimentally validated absent regulatory links in organisms, I guess, is a bit difficult to get. So that's another issue. And bringing this all together, we're thinking about, okay, how could we address this? And for me, I was thinking, okay, we need to use these additional data sets when we have them, even when they're partial, but we need to do this in a non-biasing way. So what that means is if we have gene expression often comes or is sort of genome-wide available, so we can do our networks. But if we have like binding data or other data sets that are only partially available, then we need to make sure that we do not bias towards specific stuff that we already know, recover basically what we already know. And in that context, I was thinking about using Markov random fields. And what is a Markov random field? So basically, I thought about this because my background was more like human-computer interaction and computer vision. And in a project that I did before, I was basically acquainted with them, and I kind of started to love them. And basically, just to give you an intuition from computer vision, if you take an image and in that case you tile it up into very simple rectangles here, let's call them superpixels, and then you want to do a classification on whether you see a man-made structure or you see some natural structure, as is the case in this problem here, and then basically you design your features, say, okay, that's like pre-deep learning era, you define your features and whatever, and then you train some classifiers. And what you see on the right is a logistic classifier, but basically here is takes its feature vector and then makes for each superpixel, independent of all other superpixels, classification whether this is man-made or natural. And then the idea of that Markov random field basically means that you say, okay, why not just take the environment and say, okay, if I have a strong signal in this environment for, say, natural area, and then I have this sort of classifications for man-made, which are potentially not that strong, then I might take this influence and correct my prediction for that particular superpixel. And that's actually what you see, hopefully, in this image, going from right to the left, where we have this natural area down here, and the logistic classifier, as you can see, without that neighborhood information sort of fails to classify that area on the left, where we take this into account and have these strong signals of natural regions. You see that all these white rectangles that represent man-made structure detection have disappeared. So that's basically the idea. Now it's the question, how can we sort of transfer this idea to work on gene regulatory networks? So and then we kind of came about with this GRACE algorithm, and basically I made it work so I can call it GRACE. It's gene regulatory network accuracy enhancement, and before we apply this idea of Markov random fields, first thing is that we say, okay, we need sort of a reliable initial regulatory network. So in this case, and since the system is sort of modular, you could switch out and do something else here. But what we did is, at the time, we collected transcription factor binding information and started with doing gene expression analysis, used some random forest approach, which was the GE3, which I guess most of you know from that performing very well in this Stream Challenge 2012. We used this and then we filtered this with binding information to get a bit more accurate initial network. And then we said, okay, now we want to refine this network. And what basically want to do is we want to incorporate additional data, which is potentially only partially available. We want to integrate this and say, okay, and use this idea of using neighborhood information. And so the way what we did is we kind of switched our view on the gene regulatory network, and that's kind of like the key and sort of the take-home message from the rest for the talk, if you will, is this plot B here. So what you see is we have our initial regulatory network, and then we say, okay, now we switch perspective, and we say for a single transcription factor here denoted in red, and it's potentially 2 target genes in yellow. We kind of switch this and say, okay, can we represent each link as a node in some sort of meta network? And that meta network is then supposed to represent a co-regulation. And what we then do is we build a link between these new nodes, and this is where we integrate co-functionality information. And then what we want to do is basically refine each prediction for each individual link in the original network. So the idea being, this could be a simple, I mean, it makes sense that it's going to be additional data and not the original gene expression data set, which is sort of circular. But let's say we have PPI information or something else where we say, okay, if the targets are potentially in this PPI data set found to interact, then we can use this as the link and say, okay, that gives us a bit more indicator for the co-regulation of these 2 genes. And so basically this information, if the one link to one target was very strong, thank you, then we can sort of transfer this. And okay, so basically that's what we did. And then we extract modules and we use these Markov random field idea to train this. So I have to hurry up a little bit. So I'm going to spare you guys the math. That's basically what we've been doing here, is just simply what I just summarized, finding difference distance measures. And basically the main point is that we integrate this additional data, co-functional data. Here we used like a ARANET for Arabidopsis, which we've been working with, a network that integrates different aspects, PPI and others, for co-functionality to be able to build this meta network. Okay, and that is the random field formulation. I'm going to spare you guys the details now with respect to the time, and you can find this in the paper if you want to. And basically, as I said, we built a formulation of a Markov random field, and then what we did, kind of like the random forest idea, we built an ensemble model where we finally said, okay, we have different parameters. And since that's an interesting concept that we say, okay, we have gold standard only for part of those. And we basically, what we do is we retrain the model by only taking like only a subset each time. And then we build an ensemble model and say, okay, all the links that come up robustly, they're kept. So again, this math section, you can find this in the paper. We did some tests on accuracy, compared this with other methods, and found that the model is pretty nicely with respect to both, with respect to the gene regulatory links that it could recover, as well as, and that's the interesting part, as well as gene co-functionalities between target genes. So because in our model we can optimize for both. And yeah, and then we applied this, in our case, we've been working in plant biology, we used the model plant Arabidopsis in order to ask some questions to see if we can recover links as well as we can find novel links. And here's an example where we applied this to a data set that was dealing with development, and then we could see that we could recover known regulators for cell proliferation, cell division, and also we could find additional ones. Yeah. So in summary, the idea here being that we found an algorithm that was kind of able to use additional data and sort of submodularly reevaluate individual links. If there are some links that have been weak, but when we bring in additional data, just on a submodular level, if we can still keep them and say, okay, they're still interesting, we should have a look at those. And we used for this basically a scheme from computer vision. And then we added a new optimization scheme into the system to be able to optimize for both recovering gene regulatory links as well as being able to recover co-functional gold standards. And finally, we made this into a robust ensemble model. And yeah, there's a little bit more tricks that we did, and if you want to, you find all of that in the paper. Yeah, and with this, I want to quickly just thank basically, first of all, my supervisor who've been working with at Stanford, at the Carnegie Institution, and we developed this project, and then Humboldt Foundation, who basically made it possible financially, and then of course Dream, and for being able to be here and present that work. It's pretty awesome.
Thank you, Michael. Any questions for Michael related to his algorithm? So had you worked in biology before
No, actually,
starting this?
My background, as I said, is more computer science, computer vision, human computer interaction. I think I did.
So it's really through Dream that you kind of like started this.
After my PhD, I sort of was interested in genetics, and that forward.
Great, very interesting to always have new approaches for reverse engineering. Thank you, Michael.